Assalamu alaikum everyone. So I'm back teaching you lecture 5 or topic 5 in the molecular biology part of the cytology and molecular biology course. Okay, so on this lecture we'll be covering DNA mutations. Okay, now the main resource again is the lecture itself. You can also refer to Cooper's textbook, the 11 pages shown in here, 232 to 243. These pages cover not only DNA mutations but also DNA repair. But in this lecture, in the two topics, I'd rather cover DNA mutations separate than DNA repair. I'd like you to be knowledgeable of the definitions and different types of DNA mutations before I cover DNA repair. Now, you can also refer to the NCBI website. This is an excellent resource for medical information. NCBI stands for National Center of Biotechnology Information, which is part of the National Library of Medicine, NLM, which is part of the National Institutes of Health, NIH, which is part of the US government. Now, this NCBI website or the website itself is really an excellent resource for medical as well as biological data. So it includes the complete sequence of the human genome as well as many other organisms. It's a resource for proteins, chemicals, and it has tons of information. Part of the website also contains textbooks, medical textbooks that can be relevant to you. So I suggest that you visit this website and explore it. So let's define what a mutation is first. So a mutation is a heritable alteration, but it doesn't have to be heritable. Heritable يعني وراثي. It can also include a change in the genetic material. Now, why do I say that it may not be heritable? Well, because there are two types of DNA mutations. DNA mutations can occur in somatic cells, that is, a cell in our body other than the sperm and egg, or the mutation can take place in the DNA of germline cells, that is gametes or sperm and egg. Okay, and in this case it would be heritable. Okay, so if it occurs in somatic cells, you would be the only one affected. If it occurs in germline cells, you can be affected, but your children will also be affected. Okay. Now, depending on the extent of this mutation, it can also be defined or categorized into two types: micromutations and macromutations. So what do I mean by the extent of mutations? It means how big the mutation is. So if it is a small mutation that occurs at the level of DNA, then we call it micromutation, small. If it is big, that is, it's observed at the chromosomal level. So for example, if you have an extra chromosome or if there's large deletion of a chromosome and so on, then we call it a macro mutation. Okay. So in here we will be covering micro mutations mainly. Okay. So you will have a genetics course that would cover genetic diseases and chromosomal abnormalities. Okay, so there are different types of micromutations. You have what is known as deletions. So basically in deletions, what happens is you have, let's say, a portion of a gene or a chromosome, let's say. So the same terms can also be used to define chromosomal abnormalities. So let's say that you have regions in your DNA, A, B, C, D, E, F. If you have deletion of B, well, yeah, that's a deletion, okay? Deletion mutation. In duplication, what happens is that you would have, instead of one B, you have two Bs. So the B region of your DNA is duplicated. You can have inversion, whereby you have a region of DNA that is inverted. So instead of having A, B, C, well, now you have E, D coming in place of B, C, and C, B on the other end. Or you can have translocation, What we mean by translocation is that you have a transfer of genetic material from one chromosome to another chromosome. So instead of having A, B, C, D, E, F, you would have A, B, C, then J, K, L, and this J, K, L comes from another chromosome. So this translocation can be reciprocal or irreciprocal, meaning that it can occur equal exchange of genetic material between two chromosomes, or it can be just movement of one region of a chromosome to another. Okay. So these are the different types of mutations. Now, the thing is, what does cause DNA mutations? Well, DNA mutations can occur spontaneously or they can be induced. So what we mean by spontaneously is that they happen, well, they just happen spontaneously, عشوائية, عفوية, I'm sorry. So spontaneous, they just occur, okay? It happens. Or they can be induced by something external. Like, for example, if you're exposed, if you smoke, well, then cigarettes would induce mutations. So spontaneous mutations, they just happen in cells, so they naturally occur in cells, and they can arise in all cells. Of course, many diseases can be caused as a result of mutations. Of course, primarily is cancer. Okay, so and cancer would exist almost in every single cell. Okay, except for the heart. Actually, I've never heard of cancer of the heart, which is something that is really interesting. So if you understand this mechanism, that would be really fascinating. So anyhow, so going back, so they arise from a variety of sources. They can be generated as a result of DNA replication. So as the DNA polymerase is synthesizing the DNA, well, it makes a mutation. It can create a mutation. So this is spontaneous, or it can occur as a result of spontaneous lesion. That is, something happens to the cell. Maybe you would have a certain chemical like oxygen radicals that attack DNA. Okay, so that's again a spontaneous lesion. What we mean by lesion is like an injury. Okay. Now, induced mutations are produced when an organism is exposed to a mutagen. Okay. And like, for example, if you drink عصير أبو شلن, well, that would cause a mutation. Okay. So if you smoke, well, this is also mutagenic as well. Now, some mutagens are carcinogens. What we mean by carcinogens is that they also cause cancer. Well, some mutagens are carcinogens. Now, we all know that all carcinogens are mutagens. Okay, so ionizing radiation is an excellent source for, or a common source for DNA mutations as well. UV light from the sun is mutagenic as well. Okay, so let's talk about DNA replication. What happens during DNA replication is that mistakes can happen. Okay, so the DNA polymerase can make mistakes. So you can have the most common type of mutations is what is known as base substitution. So right here, for example, you have GACG, and the DNA polymerase reads A, and instead of reading A, it puts a G instead of an A. Okay, so as a result, that would cause mutation on the other side of the strand, on the other strand, that is. So this is base substitution. We'll talk about the effect of this type of mutations later on in the translation lecture. You can also have inversion. Okay, you can have insertion. You can have deletion as well. So there are different types of mutations. So instead of GA, you would have GA, for example, and these AG are deleted. You can have insertion of, let's say, GA, another GA would be added, and then another GA, and so on. Okay, or you can have inversion, as I said. So these happen during DNA replication. Now, most commonly, what happens as well is that these mutations occur when you have triplet repeats. What we so these mutations occur at repeated sequences. So what we mean by that is, let's say, and it's really common, that you have a sequence in DNA. It's like CAG, CAG. Where are we? Okay, CAG, 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 CAG. So let's say that you have like 10 repeats. So the DNA polymerase would read CAG, 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 and then it stops. Okay, so you can imagine it stopping. Okay, so it stops and it says, how many CAGs have I added? How many CAGs have I added? So it says, I've added 10, let's add a little more. So it would add like 5. Okay, so there would be an increase in the number of repeats. Now, sometimes this increase in repeats would be harmless. It would cause nothing. But in certain cases, if you have an increase in the number of repeats, let's say from 10 all the way to like 50 or 200, and it can happen, that would cause a number of diseases. And these are some of the diseases that can be caused as a result of increasing the number of repeats. Primarily, you can have a syndrome known as Fragile X syndrome. Okay, you can have a syndrome known as Huntington's disease as well, and so on. So I don't want you to memorize these diseases and the repeats that cause these diseases, except for Fragile X syndrome as well as the Huntington's disease. Okay. What else? Well, you can have a mitochondrial condition. Okay, it's called Kearns-Sayre syndrome, and it would result from deletion of a 3-base pair repeat rather than an increase in the number of repeats. Okay, so it's different than all of these diseases that are listed in here. Okay. You can also have spontaneous lesions. That is, the DNA is damaged spontaneously inside the cells without any external factor. And there are different types of spontaneous lesions. You can have what is known as depurination. That is, D means removal of. So removal of a purine. You can have deamination, removal of an amino group. Or you can have lesions that result from oxidatively damaged bases. Okay, so let's talk about depurination first. So depurination, removal of a purine. So it's so right here you have a nucleotide that is part of a DNA strand. Remember this bond right here? It's called the glycosidic bond. Glycosidic bond. It connects the nitrogenous base with the sugar, with the deoxyribose. So what happens sometimes is that this glycosidic bond is broken. It's cleaved. Okay, so the DNA in this case would be depurinated. That is, it doesn't have a purine. And this is known as a purinic site. That is a site that does not have a purine. So what happens here is that the DNA polymerase starts to synthesize DNA. And it comes across this nucleotide. And it doesn't have a base. So what does it do? Well, remember this. It's really important for cells to stay alive. So they have a soul. So it's like no one really likes to die, right? And you would protect your soul, aggressively. So this is human nature. The same thing happens with cells as well. They do not like to die. So what happens is that when the DNA polymerase comes across such a nucleotide that is missing a base, what it does is that it stops and it says, there's a nucleotide without a base. What should I do? What should I do? What should I do? So what it does is that it adds anything. It adds any nucleotide, and it says to itself, well, you know what? Someone will fix this later on. So what's really important for me is to continue with the DNA replication, to stay alive, so that the cell would stay alive. So, and that would create a mutation, because when the DNA repair system comes to fix the DNA, it looks at it and it says, well, I don't know, there's a base that is good and there's a base that is missing. There's a nucleotide that is good and there's a nucleotide that is missing a base. So, you know what? The one that I should fix is basically I should remove the one without the base. So that would create a mutation. So there's a 75% chance that a mutation occurs, right? Because, one would be right, three would be wrong. So this is depurination. Now, deamination is something really interesting. Now, remember when we talked about the different structures of bases, and I told you, not to memorize the structures of bases, but I wanted you to, just look at the structure of cytosine, thymine, and uracil. Look at the structures here. Now, cytosine and uracil look almost exactly the same, except that cytosine has an amino group and uracil has a ketone group right here. Now, what happens with cytosine is that if it becomes deaminated, that is, you have removal of the amino group, it would convert to uracil. Okay. So what happens here is that the DNA polymerase would start to synthesize DNA, and it comes across uracil and says, that's uracil. It looks like thymine, right? I mean, thymine also has a ketone group right here, and it has a methyl group. So it says, uracil, well, you know what? It looks like a T, so I'm going to put an A. So instead of having CG, you would have UA, right? And then the uracil would be replaced by a thymine. So you would, instead of having CG, you would have TA. So that's a mutation that can take place in cells. Now, it's very common that cytosine is methylated. So right here you have the amino group and you have a methyl group. So this is methyl cytosine. If it gets deaminated, it would be converted to a thymine directly. Okay, so it's really common in our genome, in our DNA, that we have methyl cytosine. We will talk about methyl cytosine and its importance in transcription later on. Okay, you can also have adenine that can also be deaminated, and it will be converted to hypoxanthine. Now, hypoxanthine would base pair with cytosine. So instead of having AT, you would have hypoxanthine paired with cytosine, and later on hypoxanthine would be removed and replaced by a guanine, right? So you would have, instead of having AT, you would have GC in the DNA. Again, that's a mutation. All right. So mutations can also be induced. We didn't talk about oxidatively damaged bases, so it's the same concept as what we will talk about later on induced mutations. So induced mutations is basically when you have a mutation that is caused by an external factor. And you can have mutations that are caused by different mechanisms. You can have addition of a base analog during DNA replication. You can have a base that is altered chemically, and you can have a base that is damaged. Okay, so let's talk about a base analog. Base analogs are chemicals that look like nucleotides, but they're not nucleotides. Okay, and they can cause mutations. An example is 5-bromouracil. So this is 5-bromouracil right here. Now 5-bromouracil is an analog of thymine. It looks like thymine, right? So, except that what happens here is that 5-bromouracil can base pair with adenine. So the DNA polymerase starts to synthesize DNA. It looks at 5-bromouracil and it adds it to DNA, thinking that it is a thymine, right? So no problem so far because 5-bromouracil can base pair with adenine, except that 5-bromouracil can be ionized. Okay, so by itself it can just be ionized. So if it gets ionized, it would base pair with a guanine. Okay, so again, that would cause a mutation. Instead of having AT, you would have A 5-bromouracil, and then you would have ionized 5-bromouracil. Okay, and the DNA will polymerase would think that it is a cytosine because it's ionized, and it would place a guanine instead of across of the ionized 5-bromouracil. So instead of having AT, you would have G. You would have GC. Okay. Now, you can also have alteration of a nucleotide. So you can have certain alkylating agents. So what these do is that these are certain chemicals that can act that can add an alkyl group, like a methyl group right here. So this is known as 6-methylguanine. Okay, the problem with 6-methylguanine is that it pairs with a thymine. So again, instead of having GC, you would have 6-methylguanine paired with thymine, and that would result in a change from GC to a T. You can also have conjugation, attachment of large chemical adducts, chemical groups to DNA. Like in here, for example, adding this chemical here is known as benzoapyrene. Okay, it can be conjugated to guanine. Now, this chemical right here exists in automobile exhaust fumes. So all the smoke that we smell in our streets basically contains this carcinogen. Okay, this benzoapyrene also exists in cigarettes as well. So it's really quite carcinogenic. Okay, so this modification of DNA can cause mispairing, like in the case of 6-methylguanine, or it can cause carcinogenicity by damaging the DNA by distorting, that is, changing the structure of DNA, and that would cause damage to genes that can be important. Okay. Or you can have damaging DNA as a result of ionizing radiation. Okay. Now, with ionizing radiation is that you can have a lot of changes that can take place in DNA. You can have creation of AP sites, that is apurinic or a pyrimidinic site. So you have removal or break of the glycosidic bond and removal of the base. You can have base damage as well. So you can have something that looks like this. A base itself is damaged, and when the DNA polymerase comes along, DNA synthesizing it, it sees that the base is damaged and it cannot read it. So again, the cell would have a choice, either to kill itself if the DNA is not repaired, or just place anything for now and just, hoping that it would be fixed later on. Something else that can happen as a result of ionizing radiation is you can have strand breaks. So the phosphodiester bonds between bases can be broken, okay? And this is really major, and it has to be repaired. You can have single strand breaks, or you can have double strand breaks. So you can have the whole DNA would be broken at the phosphodiester bonds between nucleotides. So there are different types of damage that can take place as a result of ionizing radiation. Okay. Finally, we'll talk about intercalating agents. Intercalating agents are agents that are flat. They are composed of ring structures, and they are flat. An example is ethidium bromide. Remember, we talked about ethidium bromide before when we said that it is used to color DNA so that we can stain DNA and we can look at DNA under the UV light. Okay. Now, this ethidium bromide is carcinogenic, by the way. It causes cancer. So we don't use it anymore in our laboratories. We used to. So, you know, Allah yahmina. So anyhow, so this ethidium bromide would, the thing is that it is an intercalating agent. What we mean by intercalating agent is that it inserts itself between bases. Okay, so when the DNA polymerase starts to synthesize DNA, what it sees something and it doesn't know what that is. So again, it has a choice, either stop, and that would cause cells to die, kill themselves and die, they commit suicide. Or it says, you know what, it looks like a base, so I'm gonna add anything and fix it later. And that would cause an insertion mutations. So instead of having GTAC, right here, GAT, right here, okay, whatever, CATG, and then it would insert bases. Okay, sometimes it can insert a single base, or it may insert multiple. Okay, same thing. What it can do as well is that it says, you know what, I cannot read this, I'm gonna skip it, so it jumps over it, and that would cause deletion mutations. Okay, so these mutations are really severe if they occur within genes. We will talk about, again, deletion and insertion mutations later on. Okay, so this is the mutations lecture. A lot of information, but I think it's useful for you. But let me finish with this controversial issue, and let me raise the issue, explain it for you, and we can talk about it. So the idea here is that there is something called the three-parent baby. The idea is that we get our mitochondria from our mothers, okay? We get some from our fathers, I think, but not a whole lot. Mainly the mitochondria we get is from our mothers. So, and mitochondria have their own DNA, okay? So if the mitochondrial DNA is mutated, then babies would be affected, okay, by certain conditions. Now, so the idea here is, let's say, since, the British scientists thought, well, mitochondrial DNA is not really that important in terms of phenotypes, because the only thing that it does for cells is that it helps cells get energy. Okay, so mitochondrial DNA produces proteins that are important for generating energy, not for phenotypes. So they said, you know what? We can solve this by doing the following. We will get the egg, the ovum, from the mother that has the mitochondrial DNA mutation. We would remove the nucleus out. Okay? Now, we will take an egg from a donor female. Again, we would remove the nucleus as well. And we would place the nucleus that contains the genome of the mother that wants to have a baby into the donor's egg. Okay? And then we would fertilize this egg with the father's sperm. Okay? The husband of this female. So the idea here is that this egg would contain 3 types of DNA. The first is the nuclear DNA from the mother. The second is the mitochondrial DNA from the donor. The third, the DNA from the father. Okay. And it worked. And the baby was born healthy. And the UK Parliament approved it. And they said, this is great. Now mothers who have mitochondrial diseases can have normal babies, normal healthy babies. So the idea is now, is it halal or haram? Now the thing is that in 2016 Jordanian couple had a baby using the three-parent technique. A Jordanian couple, okay? That was interesting, huh? So this the Jordanian couple got their babies from a British developed technique. It was performed in Mexico by a Chinese American physician. Wow, globalization, huh? So here, the Jordanian newborn represents the first successful birth in new wave of so and so. So you can go back to the National Post or any other, source and read about it. So is it halal or haram? Okay, so think about it. Now I have to tell you that according to the Islamic councils and Islamic fatwa, they prohibited the technique, and they said that it's haram. Now, in 2011, I had an interview on Al Jazeera, and they asked me about it at the end, and I said, you know what, it's haram. Um, but but then I got into discussions with many people of my colleagues, and I don't know. I haven't made up my mind, actually, to be honest with you. Um, so think about it. We can discuss it if you want later on. Um, I think I'm lenient towards saying that it's halal, but it's not my opinion. But think about it, and we can discuss it later on. So enjoy, and I wish you the best.